Okay, Chapter 7, Section 5, the two-component regulatory system. Now, in prokaryotes, we're going to see that many of their responses to external stimuli are going to be under control of what they refer to as a two-component system. There's something that's going to, quote unquote, recognize a change in the external environment, which will then activate an internal enzyme, a kinase, a phosphatase, or some protein that's then, once activated, is going to go act as an activator or repressor for transcriptional activation or for transcription, expression of a gene. So you've seen this sort of similar discussed in intracellular and molecular cell biology, where you had, at the cytoplasmic membrane, you had some form of a receptor, the kinase, the kinase receptor, or a gated ion channel receptor of some sort, which would, bind to an extracellular signal and then kick off the series of dominoes, boom, 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 that were kinase signals that would transduce the signal from the receptor throughout the cytoplasm to all the organelles. Well, here with prokaryotes, it's actually a little bit simpler than that. We see that there's going to be some form of a sensor, their version of a receptor, and then there's going to be something that is transmitting that signal, some form of a signal transduction protein. As I said, these are called two-component regulatory systems. Changes in the external environment somehow trigger changes in the internal cell environment. Upregulation, downregulation of certain genes being expressed. Upregulation, downregulation of certain metabolic activities, all in response to external stimulus. There's going to be some form of a sensor kinase in the cytoplasmic membrane, not the outer membrane, the cytoplasmic membrane. So that way, this is going to be an integral protein. A portion of it's going to be touching the external environment of the cell. Then there's going to be a cytoplasmic portion that's exposed to the cytoplasm and all internal proteins and whatnot. Many times we see this is going to be something that autophosphorylates itself in response to changes in the external environment, some form of an environmental signal. And then there's going to be some response regulator. Here we're going to be looking at two-component regulatory systems that have a histidine kinase in the cytoplasmic membrane, and the response regulator is going to be a DNA binding protein that's either acting as an activator or a repressor. And with all things signaling and response, or that's, done in response to something, you have to have a way to reset the system. And we find that feedback loops are the best way. Either the ultimate product, gene product, or some intermediate will terminate and reset the regulator, reset the sensor. Usually this is done with phosphatases that will remove the inorganic phosphate group from the regulator and the inorganic phosphate group from the sensor kinase. Look, here is, a very rudimentary look at one of these two-component systems. You have the sensor kinase, quite similar to what you've been taught in other classes as a receptor. And then you have the response regulator. Does it always have to be, one response regulator? Yeah, we see that quite more often than not, that, You know, it doesn't have to be that mix of kinases and secondary messengers like we've talked about with eukaryotic cell responses that you learned in intro cell and molecular and cell biology. For one thing, we don't have all those internal organelles that all have to be, get different signals and things like that. The distance these signals have to travel through is much, much smaller, smaller cells, smaller internal environment. And also, these response regulators are going to be going directly to the chromosome, which basically fills up a large portion of the internal volume of a prokaryotic cell, both bacterial and archaea. Some environmental signal, sodium, water, whatever, binds a sensor. So sodium binds the sensor, boom. Sugar binds the sensor. The sensor changes shape, whether it's sodium, whether it's sugar, doesn't matter. Changes shape, changes its conformation, and phosphorylates itself, takes ATP, transfers that terminal gamma phosphate. Boom. It's now activated. In the process of doing that, it can now turn around and take a response regulator that's in its native inactive form and transfer that phosphate group. Signal binds, autophosphorylates, and then phosphorylates its target regulator protein. In the process, the sensor kinase resets. The phosphorylated response regulator is now in its active form, and it will go and it will bind some operator region. Okay, it's now going to act as either a repressor or an activator when it comes to transcription of a gene or an operon. Here, they're showing that the polymerase is being hindered because this response regulator is acting as a repressor. So this external signal triggers the downregulation of gene expression. Notice I keep saying turns off gene expression, because one of the things we've learned in earlier lectures in this chapter is you never truly turn on, turn off all the way. These repressors, these activators are not full 100%. Sometimes, things leak through. So that's why I keep referring to down-regulating, up-regulating. As of right now, best I can tell in the textbook and in the primary literature, there are about 50 different two-component systems in E. coli alone. So that means there are 50 different mechanisms for E. coli to upregulate, downregulate gene expression, metabolic activity in response to extracellular stimulus, extracellular signals. OMP-C, OMP-F, controlled by osmolarity, NVZ, histidine kinase detects osmotic pressure. So we can see osmolarity is a problem. That's why last slide I mentioned water or solutes in water. Nitrogen, phosphate, these metabolic activities can be upregulated, downregulated due to extracellular signals. So you go and you look, oxygen, nitrate, nitrite, organic nitrogen, inorganic phosphate, osmotic pressure. This is just a handful of the genes, the sensor kinase, and the response regulators. And here's the external environmental signal. Look, here's an artist rendition of the two-component response system in E. coli for osmolarity. You have OMP-F, OMP-C. These are porins. Okay, low osmolarity porin, high osmolarity porin, allowing stuff to pass back and forth in response to low osmolarity conditions, high osmolarity conditions. But these do not have any impact on, gene expression in response to changes in osmolarity, changes in solute concentrations. That's right here with NVZ. NVZ embedded in the cytoplasmic membrane, not the outer membrane like OMP-F and OMP-C. Changes in osmolarity, changes in osmotic pressure, autophosphorylates, ultimately leading to the phosphorylation of, what is that, OMP-R. Sorry, I don't have my glasses on. OMP-R. OMP-R acts as both a repressor and an activator. High levels of, osmolarity high, low, osmolarity high. Boom. Transcription of OMP-C. Boom. Low osmolarity. Transcription of OMP-F. How does it know which one to go to? Well, OMP-R will say yes to both. I'm sorry. Yes, it'll say yes to both, but the sequence in the promoter region, in the upstream regions, at high osmolarity, OMP-R will bind upstream of OMP-F, repressing expression. It will bind here and activate expression of OMP-C. No binding, this doesn't occur, OMP-C doesn't occur, but OMP-F will. Just OMP-R has nothing to do with that. So this seems the default, if I remember correctly, the default is expression of OMP-F. Yes, OMP-C, no. OMP-R binding stops the expression of OMP-F and preferentially goes for the transcription of OMP-C. See the same thing with nitrate metabolism. Ammonia limited, ammonia in excess. When ammonia is limited, we see that the receptor phosphorylates. Here, instead of ATP, it's using uracil monophosphate. Takes the phosphate group. NRI is a activator binding, is a repressor. Or, I'm sorry, is a activator. It binds here. What's not being shown is a bunch of other proteins that will aid in the bending of. Bends them around, and then you see the polymerase will be positioned thanks to NRI bound to the activator binding site. We get a transcription of the nitrogen assimilation gene. Under excess conditions, NRI resets, and, well, there's no reason to go through and promote uptake because there's enough ammonia coming in.